Usually when we think about artificial intelligence designing something, we picture that slightly weird image of a person with like 6 fingers, or maybe a chatbot writing a slightly generic corporate email, right? But imagine an AI model that has successfully designed a completely novel functioning virus from scratch. And this isn't, a pitch for a sci-fi movie. It's a recent, heavily documented breakthrough published in a recent issue of Nature that has sparked absolute awe in the scientific community. And, well, simultaneously, some pretty serious warnings. It really represents a fundamental shift. I mean, we are looking at a transition from AI generating human language to AI generating the literal source code of life. It's wild. I went down this Evo rabbit hole, read some pretty interesting technical architecture papers like NVIDIA's illustrated guide to Evo 2 genomic model and the hybrid transformer model called StripedHyena 2, which is a backbone of Evo 2. You know, after reading everything, I realized, wait, the whole basic concept is actually very intuitive and pretty interesting. But on the other side of the spectrum, we have these mainstream panic articles from people.com and The Guardian covering the fallout. So our mission today is to just cut through the noise, help you understand what this Evo 2 model actually is, and decode the architecture that makes it all possible. Okay, let's get into this. Before we start panicking about AI-generated viruses taking over the world, I really want to understand what this AI actually is, and what is it reading? I've seen AI read Wikipedia, but how does a computer read biology? Well, it really boils down to translating biology into a format that an AI can process. The headlines can be, very misleading because they make it sound like there's a mad scientist in a box somewhere. But in reality, you have to think of Evo 2 as ChatGPT, but replace text with DNAs. Okay? So instead of a vocabulary of tens of thousands of English words, it just has a vocabulary of 4 letters: A, C, T, and G. It operates entirely at single nucleotide resolution. But what's fascinating here is the sheer scale of the training material. Evo 2 was trained on a dataset called Open Genomic 2, which contains, get this, 8.8 trillion nucleotides. Trillion with a T. It encompasses bacteria, archaea, eukaryotes, and bacteriophages, which are viruses that specifically infect bacteria. But here is the really critical part. Nobody explicitly labeled any of this data for the model. Usually, in early machine learning, humans had to label everything like this is a picture of a cat. This is a gene. Here, they just threw the raw letters at it. Nobody told the model what a promoter region is or, where a splice site begins. It learned the statistical relationships of evolution entirely on its own through self-supervised next token prediction. It works just like a large language model learning English grammar without ever being explicitly taught the rules of syntax. Evo 2 simply looked at enormous strings of naturally occurring sequences billions of times over and just tried to guess the next letter. So over time, it just figured out that certain sequence patterns have to occur together to actually make sense. I want you to really just imagine that for a second. Try compressing the entire evolutionary history of millions of organisms into a neural network. I mean, natural evolution has spent billions of years running this massive trial and error search, finding out what keeps an organism alive and what kills it. And this AI is just quietly learning the rules of biology by reading the winning solutions evolution has already discovered. It is a profound way to approach biology. It's not just learning that a specific sequence of A's and T's does a specific thing. It is actually internalizing likelihood of those sequences coexisting in a functional genome. That's incredible. But that brings up a massive question for me. If this is essentially just an AI predicting the next token, why couldn't the researchers just use the standard ChatGPT style architecture? I mean, why didn't they just take a normal transformer model, feed the 8.8 trillion DNA letters into it, and call it a day? Well, the standard transformer architecture is brilliant for human language, right? But biological sequences expose a massive flaw in how transformers handle context. It really comes down to distance. How so? In human language, if you read a pronoun at the end of a paragraph, the noun it refers to is probably just, a few sentences away. We have local context. But with DNA, to truly understand what a gene does, you might need to connect it to a regulatory element that is located hundreds of thousands of base pairs away on the genome. So, the AI needs to be able to look at a massive chunk of DNA all at once, which means a massive context window. And that is the bottleneck. Traditional transformers use a mechanism called attention, and attention suffers from a nasty issue known as the quadratic scaling problem. Let me break that down quickly. In a standard transformer, to figure out context, every single token has to explicitly calculate its relationship to every other token in the sequence. So token number 1 asks token number 2, hey, are we related? Then it asks token 3, then token 4, all the way to the end. And then token 2 has to wake up and do the exact same thing. Now imagine doing that for 1,000,000 tokens. If you double the length of your sequence, the computational cost doesn't just double, it quadruples. If you try to feed a 1,000,000 base pair context window into a standard transformer, the math absolutely explodes. The servers would melt. It becomes completely computationally paralyzing. Okay, so the researchers hit a mathematical brick wall here. They desperately need 1,000,000 letters of context to understand the DNA. But the standard transformer just grinds to a halt if you try it. Which brings us to the architectural breakthrough called StripedHyena 2. This is the engine that actually makes Evo 2 possible. It completely rethinks how AI processes these long sequences. Let's compare a pure transformer to a very traditional librarian. You walk into a massive library and ask for a book about a company founded by someone named, let's say, Alice. The transformer librarian uses that attention mechanism we just talked about. This means the librarian literally walks down the aisles, pulls every single book off every single shelf, checks the pages for the name Alice, and then it hands you the right book. It's highly precise, but if the library has 1,000,000 books, it takes forever. Checking every single book explicitly is the quadratic scaling problem in a nutshell. It's exhausting. So, StripedHyena 2 introduces a new way of doing things. It's more like a highly sophisticated indexing system. Instead of the librarian running around checking every single page, the index groups broad themes together. It instantly knows which specific wing and which specific shelf hold books about female founders named Alice. It drastically shrinks the search area before the librarian ever has to get up. The brilliant thing about StripedHyena 2 is that it doesn't fire the librarian. It doesn't completely throw away attention. It's a hybrid architecture. It basically says, let's use the indexing system to do the heavy lifting of sorting the massive 1,000,000 token context. But let's keep the librarian around for when we need precise, explicit retrieval at the very end. But while reading the paper, I was kind of stuck on how this indexing actually works under the hood. How do they actually index a million letters without reading them one by one? Well, they do it by operating at three different distances. The researchers use what are called input-dependent convolutions. Okay, first you have the short explicit operator, or SE. This captures local context just by looking at the immediate 4 to 7 tokens. It's incredibly fast, perfect for understanding the immediate neighborhood of a nucleotide. Then you have the medium regularized operator. This widens the scope of handling dependencies over about 100 tokens. Okay, local and medium. But that still doesn't solve the million token problem, right? That is where the third operator comes in. The long implicit operator, or LI. This is a global convolution that scans the entire 1 million sequence. And the way it avoids the math explosion is by using something called fast Fourier transforms, or FFT. So I was like, wait, I know convolutional neural networks. Basically, if you want to identify a cat, then a convolutional neural network uses filter 1 for edges, filter 2 for textures, and filter 3 for shapes, right? But what on earth is FFT, which is mostly used in audio engineering, doing in a genetics AI? So, it is essentially a math trick from signal processing. Imagine a complex musical chord played on a piano. A standard transformer tries to understand the chord by measuring the exact position of every single air molecule vibrating in the room. It takes massive computing power. That sounds impossible, right? But a fast Fourier transform instead mathematically converts that complex sound wave into its core frequencies. It instantly tells you, oh, that's a C major chord. StripedHyena uses this exact same math on DNA. Instead of calculating the explicit relationship between nucleotide number 1 and nucleotide number 900,000, it transforms the entire sequence into a mathematical frequency. It grasps the global shape of the genome without doing the microscopic math for every single pair. So, it looks at the sequence as a wave, extracts the broad patterns, and skips the impossible math. You have local scanning, medium scanning, the global FFT scanning, and then voila. A layer of traditional attention just to tie the most important explicit connections together at the end. And the performance benchmark is the proof. At the 40 billion parameter scale, StripedHyena 2 achieves roughly 3 times the throughput of the most heavily optimized transformers operating at a 1 million token context. It processes massive sequences vastly faster and using way less compute. 3 times the throughput is a massive efficiency gain. But looking at that scale, 40 billion parameters, it immediately raises a question. Right now, companies are spending billions on frontier AI. We have LLMs approaching 1 trillion parameters, right? So if StripedHyena is so efficient, why not go completely ballistic? Why stop at 40 billion parameters? Why not build 1 trillion parameter biological model today? It seems like the obvious next step, especially given the whole scale is all you need mantra in Silicon Valley right now. But biological foundation models cannot be scaled exactly like large language models. There are a few very hard bottlenecks that just throwing more compute at won't solve. So, what's actually holding them back? Well, the first major hurdle is the data itself. We talked about training on 8.8 trillion nucleotides. That sounds like a lot. But unlike human text where you have, poetry, code, recipes, history, vastly different concepts. Genomes are highly conserved across evolution, meaning nature repeats a lot of the same genetic code over and over. I mean, we share a massive amount of DNA with a banana, let alone other mammals, which means the training data is incredibly redundant. It's like training a language model by having it read the exact same textbook, just tweaked slightly a million times. You hit diminishing returns on parameter count much faster in biology because the model simply runs out of novel concepts to learn. And speaking of that repetition, researchers actually altered the training laws specifically for biology. DNA repeats the exact same few letters at few places and just loop endlessly like a biological stutter. The researchers manually downweighted those repetitive sections to a value of zero point 0.1 during training. A very clever move. They essentially told the AI, do not waste your expensive computational budget memorizing this useless repetition. Pay 10 times more attention to the structural functional parts of the genome. Makes total sense. But beyond the data redundancy, there is a second, even bigger bottleneck to scaling: evaluation. Think about updating a coding AI. If you add 50 billion parameters to a Python writing model, you can test it instantly. You just run it against an automated suite of programming benchmarks, and the computer tells you in 5 minutes if the new model is better. If you update a biological model, how do you actually know its DNA designs are better? You can't just run a digital benchmark. You have to actually build the biology. You have to take the AI's digital output, physically synthesize the DNA, put it into a living cell in a lab, wait for it to grow, and observe the results. That evaluation loop takes weeks, sometimes months, and costs significant money. You can't just iterate and test a trillion parameter model overnight. The physical world slows the AI development down. Okay, so the researchers built a highly optimized 40 billion parameter engine that can digest a million biological letters at once without melting the servers. But an efficient architecture doesn't guarantee the AI actually understands biology. Did it actually work when they tested it? Let's talk about the real world results highlighted in the research papers. Well, the capabilities are genuinely stunning, both on the predictive side and the generative side. Let's look at prediction first. The major achievement here was zero-shot variant effect prediction. And zero-shot, meaning they didn't explicitly train or fine-tune Evo2 for this specific task, they just handed it a massive problem. They gave the AI the human BRCA1 gene, which is heavily linked to breast and ovarian cancer. And they introduced 3,893 single nucleotide variants of this gene, so nearly 4,000 tiny typos in the BRCA1 code. Right? Some of these typos are completely harmless, but others destroy the function of the gene and lead to cancer. And without any specialized cancer prediction training, Evo2 successfully separated the harmful variants from the harmless ones. It scored 0.87 on the AUROC scale. To put that score in context, the AUROC scale goes from 0 to 1. It is the gold standard metric for measuring how accurately an AI separates true signals from false alarms. A score of 0.5 means the AI is basically just flipping a coin. A score of 1.0 means it is a perfect oracle. Hitting 0.87 with zero task-specific training is an astonishing level of accuracy. But how does an AI trained on next token prediction actually know which mutation causes cancer? It comes back to the grammar analogy. If I say the cat sat on the refrigerator, the sentence is grammatically fine, but statistically it is highly unusual compared to the cat sat on the mat, right? You'd raise an eyebrow. Evo 2 approaches DNA the exact same way. It looks at the sequence with the typo and calculates the mathematical likelihood of that exact sequence existing in nature. Because it has internalized billions of years of evolutionary grammar, it recognized that nature would mathematically never place that specific mutated nucleotide in that specific spot. It just flagged it as bad biological grammar. That is incredible. But predicting typos is one thing. The generative side is what really made the headlines. This AI didn't just read life, it wrote it. This is where they applied a technique called beam search for guided genome generation. Beam search essentially allows the AI to explore multiple different genetic pathways simultaneously. It generates a few possible next steps, scores them all against a specific biological goal, keeps the most promising paths, and discards the rest. So it's not just randomly guessing, it's actively steering toward a functional design. They use this to design DNA sequences with specific folding patterns. But the headline grabber was the experiment with bacteriophages. They provided the AI with a known bacteriophage called Fix174 as a baseline template. They asked Evo 2 to generate variations around that biological architecture, and the AI generated about 700,000 computational designs. Out of those 700,000 digital designs, the researchers selected about 300 to physically synthesize in the lab. And 16 of them were completely viable, meaning they functioned perfectly as living viruses. In fact, when they tested a cocktail of these AI-generated phages, they successfully overcame E. coli bacteria that had naturally evolved resistance to normal bacteriophages. It designed a novel biological weapon against a resistant bacteria. Now, this is exactly where the terrifying headlines in People magazine and The Guardian come from. So, what does this all mean? If it can design a virus to beat E. coli, are we essentially handing a custom pathogen generator to the general public? It is a very critical concern. First, it is important to state that the researchers intentionally implemented safety guardrails. They did not train Evo 2 on any viral genomes that infect humans or eukaryotic cells. Okay, so it literally lacks the vocabulary to write a human virus. It only knows how to infect bacteria. But the proof of concept is out there now, which is exactly why experts are divided. On one side, you have biosecurity experts from Johns Hopkins who are urgently calling for new global governance. Their position is that the foundational capability to compose viral genomes using generative AI now exists, but the regulatory frameworks to safely monitor it absolutely do not. They're arguing that the cat is out of the bag and we are just legislatively blind. They are deeply concerned about the unpredictable nature of entirely novel pathogens generated by black box algorithms. However, on the other side of the debate, you have synthetic biologists like Tom Ellis from Imperial College who publicly call the threat, quote, overblown. So, how can generating novel viruses be an overblown threat? Well, his argument focuses on the practicality of bioterrorism. If a bad actor wants to create a devastating human pathogen, asking an AI to design a complex genome completely from scratch is incredibly difficult and highly prone to failure. Nature has already evolved perfect, highly transmissible chassis like influenza or COVID. So instead of inventing a new biological engine from scratch, a bad actor just tunes up a sports car that already exists. It is vastly easier and more reliable to take a known human pathogen and make minor targeted gain of function tweaks to its receptors to make it more lethal. You don't need a massive foundation model to do that. And this leads to the consensus view on security articulated by Dr. Philippa Lenzos from King's College London. She points out that obsessing over the AI software misses the actual choke point, the physical printing of the DNA. You can generate 1 million deadly genomes on your laptop, but they are just code. To make them real, you have to send that sequence to a DNA synthesis company to physically manufacture the genetic material. That physical synthesis choke point is where stringent screening, customer verification, and biological safeguards must be enforced. The synthesis companies need to heavily scrutinize what they are printing before they ship it in a vial. For me, that makes perfect sense. Okay, so we have covered a massive amount of ground today. We started with the sheer scale of an AI predicting the literal alphabet of life from 8.8 trillion letters. We explored how the StripedHyena architecture bypassed the standard transformer bottleneck using fast Fourier transforms to grasp the global shape of 1 million letter sequence. And we saw how that efficiency allowed a 40 billion parameter model to correctly diagnose cancer, causing mutations, and generate physically viable viruses. It is a true milestone in our understanding of biology. But looking at the architecture itself, this raises an important question, one that extends far beyond genetics. We have spent this entire time discussing biological applications, but striped hyena 2 is a generalizable architecture. If this system can successfully and efficiently index 1 million token contexts in the chaotic, highly repetitive world of DNA, what happens when this hybrid architecture inevitably might migrate back to human language? We might very soon be interacting with language AIs that don't just reference a few recent documents. We are looking at a future where an AI can ingest, retain, and flawlessly cross-reference entire libraries of human history, decades of complex legal precedent, or the entirety of global medical literature, all within a single prompt without the servers melting down. The ultimate indexing system for the entirety of human knowledge. Thank you so much for watching. There's clearly a lot more to explore here, both in the biology and in the future of AI architecture. So keep asking questions, keep exploring, and I will see you in the next video.